Male sterility is one of the techniques to avoid the self-pollination and promote crossing. Male sterility can be classified as genetical and non-genetical. In non-genetical method, chemical hybridizing agent is the primemost. This is also popularly called as gameticides, and in this video we shall discuss about that. To maximize any hybrid seed production, there are certain criteria like source of viable pollen from a male parent that can outcross with male sterile, that is a female parent. Female planting configuration that will maximize outcrossing. Agencies such as wind or insect to move pollen from the male to the female parent should be there. Female and male parents that are in synchrony, that is pollen from the male parent must shed, then male fertile, male sterile, female parent is receptive. Abundance of pollen from the male parent must be present. Good flower opening characters in the female parent. All these should be the characters to maximize the hybrid seed production. Either it may be male sterility, self-incompatibility, whatever it may be, but especially in case of male sterility, either it may be genetic or non-genetic. So now the chemical induced male sterility or gameticides. This is a non-genetic male sterility system. Term chemical hybridizing agent was suggested by Macrae in the year 1985 to avoid confusion and multiplicity of terms like male gameticides, male sterilants, selective male sterilants, pollen sterilants, then pollen represents androcytes, etc. The first report was given by Murray and Naylor in the year 1950. They induced male sterility in maize using malic hydrazide. In 1951, 1 year after this discovery, Lebeck and Cribben reported that Na, that is naphthalene acetic acid, and beta indole acetic acid, these both are auxins. This increased the proportion of staminate flowers in cucurbids. Staminate means the male flowers in cucurbids. If you will apply ethophone, that may lead to increasing the female flowers in the cucurbids. Like that, hormones or artificial plant hormones, it may be artificial or natural, play a very important role in determining the male organs or increasing the number of male flowers as well as female flowers. Sterility induced by application of certain chemicals, like categorization of chemicals are auxins and antioxidants. Example NMTBA 240 or malic hydrazide, etc. Then halogenated aliphatic acids include FW450, dalaphone, gibberellic acid like GA3, ethophone, OPX 3718, arsenicals like MSMA, OM, ZMA, etc. Along with that, RH531, RH532, etc. are some of the categories. These chemicals are called gameticides since they lead to pollen abortion and cause male sterility. Sometimes it results in female sterility also. So the term chemical hybridizing agent are used since after the entire primary objective is to produce a hybrid. More than female sterility, male sterility is given much priority. Number of chemical hybridizing agents have been reported to cause male sterility for producing the hybrids. Therefore, no need to be developed maintainers or restorer lines, and this can help in saving time, labor, and money. Only thing is, we should know the biological process of individual species and act accordingly. The exact stage of application of any chemical is known, then the hybridization program will be a great success. Coming to the properties of ideal chemical hybridizing agents. The CHS or CHAs, or we call it as gameticides, must be highly male or female selective, means they should be specific to male or female, not to both. Should be easily applicable and economic in ease. Time of application should be flexible, must be must not be mutagenic, must not be carried over in F1 seeds, means they should not be heritable, must consistently produce more than 95% of male sterility, must cause minimum reduction in seed set, should not affect outcrossing, should not be hazardous to the environment. Some of the important chemical hybridizing agents I have listed here, like zinc methyl arsenate, popularly called as Mg1, and sodium methyl arsenate, called as Mg2. These should be applied 5 days before heading in rice. That means arsenical chemicals like zinc methyl arsenate and sodium methyl arsenate are specific only to rice, and this must be applied 5 days before the heading process. Second is ethefone or ethyl. This is popular in barley, oat, bajra, rice. The dose is 750 to 4000 mg per liter or 0.2 to 1.2 kg per hectare. The the stage of application or critical stages depends on the crop, because it is applicable in many crops. Then mentic is applicable in cotton and sugar beet. The stage of application also depends on crop. Gibberellic acid, this is used in maize, barley, wheat, rice, sunflower. Application is 1 to 3 days before meiosis process. Then LY195259 is specific to wheat, and the dose of application is 1.12 kg active ingredient per hectare. Then W84811, popularly called as shell, is applied at booting stage, popular in maize, barley, wheat and oats. 1.2 kg per hectare is the active dose. Dolafan, this is sodium 2-dichloropropionate. This is popular in cotton, pearl millet, wheat, linseed, sesame and onion. Then DPX 3778, that is 3p chlorophenyl 6 methoxy 3 thiazine 2,4 dione. Useful in maize, wheat, triticale. Then malic hydrazide 1,2-dichloropyridazone 3,6-dione. Malic hydrazide is common in maize, wheat, capsicum, cotton, oats, sorghum, onion and okra. Mentic is FW450. This is popular in capsicum, cotton, pearl millet, sesame, sunflower. The chemical name is sodium alpha beta dichloroisobutyrate. Then Hybrex, it is RH0007. The dose is 0.2 kg per hectare to 2 kg per hectare. 2.2 kg is for spring wheat and 2 kg is for red winter wheat, and it is popular in wheat. Now, ethofone dose, as I told you, it varies from crop to crop. So we shall see here. For barley, the dose is 700 to 1500 mg per liter. Stage of application is late boot or penultimate leaf appearance or early boot stage. In mustard, the dose is 2000 to 3000, that is applied just before bud initiation. In bajra, 2000 mg per liter, applied at late boot or early protogyny stage. In rice, 1000 to 3000 mg per liter, that is applied at early boot stage, and in wheat it is 1000 to 2500 applied at pre-boot stage. So how the chemical hybridizing agents are used in hybrid seed production? The conditions required for this are proper environmental conditions like rains, sunshine, temperature, relative humidity should be optimum. Temperature must be between 26 to 28 degrees Celsius, whereas relative humidity of 80% or more. Then synchronization of flowering male and female parents, like isolation should be practiced in order to synchronize the flowering habit, maybe spatio or temporal. Effective chemical emasculation and cross pollination should follow. Precise stage and with recommended dose must be practiced. Gibberellic acid spray to promote stigma exertion, then supplementary pollination to maximize seed set. Avoiding chemical hybridizing agent spray on pollinator row. 2 doses of chemical hybridizing agent in lower dose is better than high dose at a single dose. That means proper stage, amount, then particular chemicals used, its specific features, chemical nature, and everything must be known. Along with that, if the doses are given in n number, instead of giving it for a single time on high dose is preferred. Utility of chemical hybridizing agents: high performing female parents on F1 hybrid. That means female hybrids are promoted by emasculating their male non means which are not preferred one. Then it can use where emasculation is difficult. That means gameticides are helpful where emasculation process is very difficult, like small millets. In millets, the flowers will be very minute. Even in pulses, the flowers are delicate, so that wherever handling is difficult, emasculation is easy by using gameticides, especially in self-pollinated crops. Any line can be used as a female parent here. No need to maintain ABR line. Choice of parent is entirely flexible. Chemical hybridizing agent based F2 are fully fertile as compared to few sterile hybrids in case of CMS or GMS. It's also provide ideal material to study for the productive growth, cytoplasmic inheritance, and pollen development. Coming to the mode of action of gameticides means on which biological process it affects, we shall see here. Disruption of meiosis leading to arrest of anther development and degeneration of pollen mother cells or curly microspores. Disruption of exine formation result in thin-walled, irregular or persistence and abnormal growth of tapetal layer, decrease of starch deposition and viable microspores, appearance of abnormal vacuoles in the microspores making them non-viable, indehiscence or delayed dehiscence of normally developed anthers with viable pollen, non-germination of pollen on stigma or cessation of elongation of pollen tube resulting in non-fertilization. The stages of treatment, it depends on the nature of damage. Here, the two things are treatment at panicle initiation stage, that is pre-meiotic effects with normal development of stamens and primary mother cells. The treatment at bolting stage, that is meiotic phase, and flowering stage is post-meiotic, disturbs normal development and functioning of the microspores. Any disturbance to plasmodesmata which connects sporogenous and tapetal cells may lead to non-development of primary mother cells. Once PMCs are formed, tapetum plays significant role in causing sterility, overgrowth of tapetum in terms of either formation of double layer or clone acting action or tapetal cells prior to callus formation of primary mother cells lead to crushing of primary mother cells. Blockage is synthesis of calluses may also render the primary mother cells sterile. All the foregoing result of pre-meiotic disturbance. Once the development process reaches meiotic phase, treatment effect is reflected in the form of varied cytological anomalies like chromosome breakage, irregular pairing, orientation and dysfunction of chromosomes, occurrence of laggards, and uneven disturbance or distribution of chromosomes, all affecting normal development of and viability of pollens. Here you need to concentrate here where the chemical hybridizing agents particularly work. They work on primary mother cell, they disturb the meiosis. As a result, cytological differences are observed like chromosome breakage, irregular pairing, orientation, dysfunction of chromosomes, occurrence of laggards, uneven distribution of chromosome. Then post-meiotic disturbance, starting from tetrad stage, extend till dehiscence of anthers and release of pollen. Hindrance to microspore separation during tetrad stage, causing sterility is due to lack of callus formation. Then chemical specificity disrupting the callus synthesis induce sterility through this phenomena. Tapetal cells surrounding the developing pollen cells play essential role in the development of pollen grains by serving the nutritional channel. So what will happen? The chemical hybridizing agents disrupt the this particular tapetal layer. These are the pollen grains, four pollen grains, and the outer portion is the tapetal layer. So tapetal layer provides the nutrition, and if the gameticides are working on the tapetal layer, that means pollen growth can't be possible. At the end of interphase, microspore nucleus divide unequally, forming vegetative and generative cells. This division being a very unusual type, it becomes vulnerable to chemical hybridizing agents which are specific to post-meiotic phase. That means chemical hybridizing agents are again categorized as pre-meiotic and post-meiotic and certain stage specific. The generative cell divides once more in the pollen grain, forming two sperm cells. Normally the generative cells is near the intine wall, and if any chemical renders it impossible either to be at that place or to divide, the pollen becomes sterile. The utility of chemical hybridizing agents in wheat. Here why I am focusing on wheat, because in other systems, two line system, three line system, maybe cytoplasmic male sterility or genetic male sterility or cytoplasmic genetic male sterility are harnessed in many of the cereals, but wheat itself is a hybrid, so application of the genetic male sterility is quite difficult here because of the complex genome structure. So that chemical hybridizing agent is the one which is widely used in wheat. Even it is used in rice also, but here I am talking about wheat. By using chemical hybridizing agent, emasculation of each floret is difficult. Low seed multiplication per this one, and non-availability of effective ms system is a problem in wheat. But using the chemical hybridizing agent, there is an increase of heterosis ranging from 3.5 to 15% is observed. So one more new chemical is introduced, that is SQ1. It's a pyridazine compound. Main ingredient is 4-chloroniline. Highly effective for utilizing the heterosis. Very low phytotoxic and pollution. Can lead to complete male sterility. No side effects on agronomical trials. So how the SQ1 mode of action takes place? We may apply it on anther or leaf. What will happen? The component of ribosome, DNA metabolism, or aerobic respiration, photosynthesis, reactive oxygen species metabolism, anaerobic respiration, glutathione metabolism, abnormal RNA degradation, protein degradation. Some of these kind of problems will be faced by the plants, and ultimately it leads to male sterility. That means we should not apply it on the leaf. If we apply it on anther, well and good, it will induce the male sterility. Coming to the advantages of chemical hybridizing agents, any line can be used as female here. Choice of parent is flexible. Then this can be used as two line system. Maintenance of parental line is possible by self pollination. Based F2 hybrids are fully fertile as compared to few sterile hybrids in case of CMS and CGMS. Coming to the limitations, gameticides cannot be utilized for a large scale seed production, used only to produce sufficient seeds for the breeding program. Because the application will be very difficult, then it may cause severe effect on the environment. Even it may lead to the costly approach. Then expression and duration of chemical hybridizing agent is stage specific, so that skilled labor as well as knowledge of particular plant is very essential. Sensitive to environmental conditions, like if temperature and relative humidity is varying, that means again the chemical application will be impaired. Then incomplete male sterility produce self-fed seeds, again it will be a failure of male sterility. Then many chemical hybridizing agents are toxic to plants and animals, possess carryover residual effect in F1 seeds. That means residual effect even may enter our ecological chain, interfere with cell division, affects human health, genotype, dose, application, and stage specific. Friends, the information furnished by me, I think it is helpful to you. If you like this video, please do not forget to like, share, and subscribe my channel. Thanks once again for patience listening and watching.